Okay, I want to thank the organizers for giving me this chance to speak in that very kind introduction. The title of my talk is In Vivo Delivery of Transcription Factors with Multifunctional Oligonucleotides. I'll actually talk about two projects in this presentation. The first will be on delivering transcription factors, and the second project will focus on targeting bacterial infections for imaging. So transcription factors play a central role in biology and medicine, and there are numerous transcription factors that could have tremendous therapeutic power if they could be delivered into cells properly. We have focused on delivering one particular transcription factor called NRF2 as a potential therapeutic, and this is because of NRF2's central role in protecting cells against oxidative stress and any type of inflammatory disease. The biology of NRF2 is shown on this slide here. NRF2 is it's a transcription factor that's normally complexed with a protein inhibitor called KEAP1. So NRF2 is made by almost every cell in our body, but it's inactive in most cells because KEAP1 complexation causes NRF2 to be ubiquitinated and then degraded in the proteasomes. However, in the presence of any type of oxidative insult or any type of inflammatory process, KEAP1 gets degraded. That allows NRF2 to go to the nucleus and cause the transcription of about 300 genes that have been demonstrated to protect cells and animals against almost every type of inflammatory disease, ranging from Alzheimer's to atherosclerosis. There's therefore great interest in being able to deliver NRF2 into cells and convert it into a therapeutic. However, delivering NRF2 is challenging because NRF2 is a transcription factor, and transcription factors are very sensitive to chemical modification and also to organic solvents, and therefore traditional delivery approaches have not been very effective. We have developed a strategy to deliver transcription factors into cells it's called the DARTs, and the DARTs stand for DNA Assembled Recombinant Transcription Factor Delivery. The molecular design and chemical structure of the DARTs are shown here. So the DARTs are composed of an oligonucleotide sequence that contain the promoter sequence of the transcription factor we want to deliver. So the idea is that this is a 20 to 40 base pair oligonucleotide sequence that happens to have a high affinity of the transcription factor because it's part of the promoter sequence. So in this case, we want to deliver NRF2, and this is the NRF2 promoter sequence. So this part binds NRF2. We have then chemically conjugated an endosomal disruptive agent, which is essentially this large hydrophobic region, to the DART, to this oligonucleotide, and we've also connected two galactose units for liver targeting. The idea is that we can just take this molecule here, which we can easily make, complex it to our transcription factor, it binds here, we then inject it, this part triggers endocytosis, and then we also have an acid degradable linkage between this hydrophilic targeting group and this hydrophobic membrane disruptive group. Once after endocytosis and in the endosome, this part hydrolyzes, exposes this domain, and allows the transcription factor to get into the cytoplasm because this unit is supposed to disrupt the endosome. How this is all supposed to work is again shown on this slide here. So the DARTs are composed of this DNA that binds the transcription factor. Here I'm showing the transcription factor, that's the NRF2. So we make this complex by simply making this molecule and mixing it with NRF2. We then inject it. This part, which is the galactose groups, binds the galactose receptor on the hepatocytes. That triggers endocytosis. But once in the endosome, the pH goes down to 5. This acetal linkage, which is acid degradable, hydrolyzes. That exposes the hydrophobic domain, and this hydrophobic domain can then penetrate the lipid bilayer and release NRF2 into the cytoplasm. So the DARTs are designed to be pH-sensitive membrane disruptive agents because they contain an acetal linkage that is designed to hydrolyze at acidic pHs. We therefore measured the hydrolysis of the DARTs at both pH 5 and pH 7.4. We did this by incubating the DARTs at 2 different pH solutions and doing electrophoresis. The results of this experiment are shown here. The y-axis is percent hydrolysis. The x-axis is time in minutes. And what you can see is that at pH 5, the DARTs have a half-life of about 1 hour, whereas at pH 7.4, the hydrolysis half-life increases to about 24 hours. And this is expected based on the acid sensitivity of the acetal linkage, and also means that that the DARTs have hydrolysis kinetics that's suitable for endosomal disruption. So after endocytosis, they should be hydrolyzed and start disrupting membranes within 1 hour, and that's about the time scale of trafficking the lysosomes, so they should have not too much degradation in lysosomes, but a 24-hour hydrolysis at 7.4 means that they should have enough time to circulate in the blood and find the liver. We therefore measured the membrane disruptive properties of the darts at both pH 5 and pH 7.4. We did this using a calcine liposome leakage assay. In this assay, we make liposomes which have a very high concentration of calcine in them, and the calcine is therefore self-quenched. We then take these calcine-containing liposomes, incubate them with the darts at pH 5 or pH 7.4, and measure the resulting fluorescence, which represents membrane disruption. The results are shown here. The y-axis is percent calcine leakage, the x-axis is dart concentration, and what you can see is that the darts are about 2 to 3-fold better at disrupting membranes at pH 5 than they are at pH 7.4. We therefore investigated if the darts could alter the intracellular distribution of NRF2 that they deliver. To do this, we took NRF2 and labeled it with CY3, and then complexed it without the darts, added them to HepG2 cells, and then co-stained these cells with lysotracker green, and then did confocal microscopy. So on these slides, the green stain is the lysosome or endosome, and the red stain is the NRF2. What you can see is that in cells just treated with NRF2, you see a lot of co-localization of NRF2 with lysotracker green, and that shows up as yellow. So these yellow spots here are direct co-localization, but you see very little NRF2 that's by itself. And most of it's co-localized in lysosomes. However, if you take NRF2 complex into the darts and deliver that, you do see a lot of red that's away from any type of green fluorescence, suggesting that the darts are able to alter the intracellular distribution of NRF2, presumably by endosomal disruption. Okay. We therefore investigated if the darts could deliver NRF2 and cause the transcription of NRF2 downstream genes in cells. To do this experiment, we took darts, complexed them with NRF2, added them to hepatocytes for 4 hours, and then washed everything off, waited for 24 hours, and then made cell lysates of these cells and did RT-PCR of 3 NRF2 downstream genes. These are heme oxygenase, NQ01, and GCLC. The results are shown here. And what you can see is that the NRF2 complex to the darts does cause a dramatic increase in heme oxygenase and also NQ01 and GCLC. This is about a 3 to 5-fold increase, whereas free NRF2 has no effect. Okay. We have done a series of in vivo experiments to investigate if the darts can enhance the delivery of NRF2 in vivo. We used an acute liver failure model for these studies, and in this animal model, what we do is we take mice, give them an overdose of Tylenol, wait for 1 hour, and then do a tail vein injection of NRF2 complex to the darts. We then wait for 24 hours and measure ALT levels in the blood. So what's supposed to happen is that the darts, this NRF2 should get complex to the darts, get endocytosed in the endosome. This part should hydrolyze off, exposing the membrane disruptive domain, and allow NRF2 to go into the nucleus and cause the transcription of downstream genes. So the results of this animal experiment are shown here. The y-axis is the various experimental groups. APAP is the same thing as Tylenol, and the x-axis is serum ALT levels, which is a surrogate marker for liver toxicity. What you can see is that mice just treated with Tylenol or APAP by itself have about a 7-fold increase in liver toxicity. Free NRF2 has no effect, but if you take NRF2 and complex it to the darts, you get a dramatic reduction in the toxicity, and it's essentially statistically not different than the control. We've also been able to verify that we do see downstream NRF2 genes expressed. We are now looking at delivering NRF2 in more complicated animal models of liver disease, such as liver toxicity caused by tuberculosis drugs. Okay, I want to switch gears now and talk about another project in our lab, and this project focuses on imaging and targeting bacterial infections. So bacterial infections cause millions of deaths each year, despite the availability of antibiotics. One of the major challenges in treating bacterial infections is diagnosing them at an early stage. At present, bacterial infections are diagnosed either via a blood culture or a tissue biopsy. However, both of these methods can only detect late-stage infections that are challenging to treat. There's therefore great interest in being able to develop new strategies for imaging and diagnosing bacterial infections. we have identified the maltodextrin transporter as a target for imaging bacterial infections. The maltodextrin transporter has 3 properties that make it ideal for targeting bacterial infections. The first one is that the maltodextrin transporter is only expressed on bacteria. It's not expressed on mammalian cells. So this strategy is very specific for bacteria. The second one is that maltodextrins, which are glucose oligomers 2 to 12 units in length, are the preferred food source for bacteria. So bacteria internalize maltodextrins at an extremely rapid pace because they use it as a food source and essentially a food storage source. They basically take these oligosaccharides, break them down into individual glucose units, and use that glucose as a food source. So bacteria internalize these at an incredibly rapid rate. And the final one is that the maltodextrin transporter is fairly promiscuous. It only seems to recognize, at least in E. coli, the first 2 glucose residues at the non-reducing end. And it doesn't seem to care about what's on the other end of this molecule, which is the reducing end. And so that allows us to conjugate almost anything to the reducing end of these oligosaccharides and get them into bacteria with high selectivity in high quantities. So we therefore developed a strategy for targeting bacteria initially for the sake of imaging by conjugating imaging agents to the reducing end of maltodextrins. The first compound we made to investigate this strategy is called MDP1. That's composed of parylene conjugated to maltohexose. We made this via click chemistry, hence the triazole ring. We then incubated it with 4 common pathogenic bacteria: E. coli, Pseudomonas aeruginosa, Bacillus subtilis, and Staphylococcus aureus. And we did this at a 10 micromolar concentration and waited for 1 hour and then looked at the intracellular fluorescence. The results are shown here. The y-axis is millimole concentration of MDP1 per CFU. The x-axis is the various bacteria. And what we see is that all the bacteria, E. coli, Pseudomonas, Bacillus subtilis, and Staphylococcus aureus, have millimolar concentrations of this compound, even though they were only incubated with a 10 micromolar concentration. And it turns out that we can actually get about 50 millimolar or 100 millimolar concentration of compounds into bacteria, in vitro at least, using this strategy. We've done a variety of in vivo imaging experiments to investigate if we can image bacteria in vivo targeting the maltodextrin transporter. To do these experiments, we made this compound called MDP2, which is composed of the dye IR786, a near-IR dye conjugated to maltohexose. We made this dye again via click chemistry, conjugate, and did animal experiments where we injected 10 to the 7 E. coli into the thigh muscles of rats, did a jugular vein injection of this compound, and then after 16 hours, which is about the time it takes for this compound to clear, we can see very high localization in the area of the infection and almost no localization anywhere else, suggesting that MDP2 has very high specificity for bacteria and that its internalization by mammalian cells is very low even in vivo. Okay. One of the nice things about targeting the maltodextrin transporter is its large tolerance for modifications of its substrates. This allows us to deliver a wide variety of things to bacteria. We are very interested in being able to image bacterial infections by positron emission tomography. This is because PET imaging agents have a very rapid translational pathway into the clinic and because of the widespread clinical use of PET. To do PET imaging of bacteria, we made this compound here, which is F18 conjugated to maltohexose. We do this chemistry by first taking this azide-protected acetate, this azide maltohexose derivative, which has all its hydroxyls protected as acetate groups. We click on this broselate intermediate. That gives us this compound here. We then give this to our collaborators who are at Emory University, and this is the PET chemist Mark Goodman. So we give this to the Goodman laboratory, and they do a one-step F18 labeling with potassium fluoride. And they do it at about 110 degrees in 1 hour, and we can now reproducibly get radiochemical yields of about 4.5%. And so we do the fluorine displacement on that carbon there, and also the deprotection all at the same time. The Goodman Laboratory has done numerous experiments with MHF to investigate if it can image bacteria in vivo with specificity. One of the first experiments they did was they injected 10 to the 9 E. coli into the tricep muscle of a rat and then imaged with this compound here. And so this is 1 hour after injection. You can easily visualize the region of the infection, and this compound seems to clear from all the other organs fairly well. Based on these studies, we then did a side-by-side comparison comparing the ability of MHF to image bacterial infections versus FDG, which is fluorinated glucose. That's essentially the industry standard for PET imaging. So to do this experiment, we took 10 to the 9 bacteria, injected them into the left tricep muscle, as shown there, injected either FDG or MHF, and then after 1 hour, we sacrificed the rats into the biodistribution study. The results of this experiment are shown here. The y-axis is % injected dose per gram of tissue. The x-axis is the various organs. The red bar is MHF. The blue bar is FDG. And what you can see is that in the infected mice, we see about a 41-fold difference between infected versus uninfected muscle, whereas with FDG, there's only about a 2-fold difference. And most importantly, MHF clears from all the uninfected organs extremely well, especially in the heart, the lung, and the brain, whereas FDG has a lot of accumulation in these tissues. So we think MHF will be much better at imaging bacterial infections than FDG because of its low nonspecific accumulation. We've also looked at the minimum number of bacteria we can investigate. We can go down to about 10 to the 5 E. coli, and that's orders of magnitude better than other PET imaging agents. And one other thing we can do with MHF is actually measure bacterial drug resistance essentially in real time. And this is because the maltodextrin transporter needs ATP to function, and only metabolically active bacteria internalize it. And we can therefore distinguish drug-resistant bacteria versus wild-type bacteria, because the drug-resistant bacteria will survive antibiotics and will still be taking up the probe. So in conclusion, I've talked about 2 new technologies. The first one was the DARTs, which were designed to deliver transcription factors. The second one was the MDPs, which were designed to image bacteria. One other aspect of the MDPs that we're now exploring is using it to target antibiotics to bacteria, and I'd love to discuss this with people or collaborate with people about this. And finally, I want to thank the people that did the work. The NRF2 project was done by one very talented graduate student named Kunwoo Lee, and the MDP project was done by two postdocs. One's name is Xinghai Ning, who's now a professor at Nanjing University, and Xiaojian Wang. I want to thank the NIH for funding. And finally, I'd like to thank you for your attention, and if there are any questions, I'd be happy to answer them.
Thank you, Niren. Any questions?
It's really nice talk. I just have one question about the first part.
Yeah.
I think you test in the messenger RNA level by RT-PCR.
Yeah.
Did you test in the protein level?
Yeah, great question. So, yeah, we have done Westerns, and we do see increases by Westerns also, yeah.
Yes, really nice talk too. Have you tested the DART without the NRF2?
Yeah, great. Yeah. So for in vivo, we have looked at and we don't see any. We see no statistically significant differences in. Yeah.
Is there any differences in complexing NRF2 with KIP? Is there a displacement?
We don't know. We don't know yet. Yeah. We don't know. Yeah.
Even in vitro.
Even in vitro, we don't know. We don't. Yeah. That's.
Any other questions? Could I just ask, have you been able to target the DART to other than the liver? Have you looked at other tissue targeting?
Yeah, that's a great suggestion. We haven't yet. Yeah.
And the stability of the DART, it seemed that at 7.4 it did have some instability. Is that going to be compatible with clinical formulation? Do you think of these agents?
Yeah. So we haven't seen a lot of toxicity with the DARTs in vivo, even at high concentrations. Yeah, we're still not yet. Yeah, so I think in cell culture, hundreds of micrograms per ml, we're not seeing any toxicity. So I think it turns out that the calcine liposomes are a lot easier to disrupt than mammalian cells probably is where.
Okay, well, yeah. Thank you very much. I think we should draw things.